Hi, in this video we're going to talk about eukaryotic gene structure as well as the structure of messenger RNA. Alright, so let's start with a region of DNA. This is going to be somewhere on one of the chromosomes, and we're going to represent it in this video with this gray rectangle. So do keep in mind this is representing double-stranded DNA, and we're going to highlight some of the sequence features. All right, so let's start with some of our regulatory elements. And we're going to start with enhancers and promoters. So the promoter will be the region where RNA polymerase binds. It's just directly upstream of the part of the gene that will be transcribed. Now, enhancers are going to be a little bit further away from the region of the gene that will be transcribed. I represented them here as upstream of the promoter, and they often are upstream of the promoter, but they also could be downstream. They could be in an intron. They could even really be on another chromosome. They could be very far away from the actual gene. And enhancers are DNA sequences that are regulatory. Sometimes we even call them regulatory switches, but the name indicates that they enhance transcription. Now, let's say that they are upstream. The way that they're actually going to have any effect on this gene is that the DNA is going to bend to bring the enhancer and promoter into close physical proximity. And then these blue rectangles represent activator proteins. These are DNA binding proteins that will bind certain sequences in the enhancer, and they're going to enhance transcription by helping to recruit transcription factors. So these are represented by the colored ovals. These are proteins that bind either to the DNA of the promoter or they bind to other transcription factors. They sort of assemble as this complex, and they recruit RNA polymerase to this promoter region. All right, so now we're teed up and we're ready to start transcription. All right, here is the region of the gene that is transcribed, and I've color-coded this into blue, green, and yellow. So let's talk about each of these, starting with the blue. So the blue represents untranslated regions. So these are regions that will be transcribed, so they'll be part of the messenger RNA, but they're not containing information that will be directly translated by the ribosome. So we have two of these, the 5' UTR and the 3' UTR. 5' UTR has regulatory regions, some of which might indicate to the ribosome to start translation. 3' UTR also has regulatory regions. One important function of that is to control polyadenylation, okay, or in other words, the addition of a poly A tail to the messenger RNA. Now, in addition, we have in this transcribed region exons and introns. Exons are represented here in green. Exons are the part of the gene that are actually going to be translated. So these contain protein coding information. You can think of exons as they're going to exit the nucleus and actually go and interact with a ribosome, as opposed to introns. So these are intervening sequences that need to be removed from the transcript before translation. You can sort of remember them as staying in the nucleus, because remember, we're talking about a eukaryote here. All right, so we have our exons, our introns, and our untranslated regions. And that is going to give us our primary transcript. This is going to be a molecule of pre-mRNA. Now, messenger RNA needs to be processed before it leaves the nucleus and is translated. And there are sort of three distinct things that are going to happen to get our mature messenger RNA. So we're going to add something to the 5' end as well as the 3' end. On the 5' end, we add a cap. This is going to help protect the messenger RNA from degradation. And on the 3' end, we add that poly A tail. So when I refer to the 3' UTR as controlling polyadenylation, or at least regulating it, this is what we're talking about. So we just add a long sequence of adenines. And once again, this will protect our transcript from degradation. We also splice the introns out. So you can see that those yellow sequences have been removed. And now we're left with just the exons. In this case, we have 5 exons, although there could be fewer or many more in a eukaryotic gene. And these exons are going to be the coding sequence that the ribosome will actually translate and use to make a polypeptide that can then be folded into a functional protein.